I am Dr. Asit from India, and Calcutta is my city where I live, and my university was away in the west about 100 miles, so that is very much village-like. It is called Medinapur city. So, well, I have retired recently, and I will talk on Phyto-Drugs. And the aim is to isolate the abundant phytochemical that inhibits MDR E. coli bacteria. And overnight ethanol extract of plant part was used because water extract mostly no antibacterial activity. That is my findings. And my system is 40 microliter of a extract if give 50 millimolar of the diameter of the lysis zone in a Kirby-Boyer assay, then I thought it could be proceed because that is significant industrially. Preparative TLC was made and purification done 4 times, and we characterized spectroscopy method was used. Indian Phyto-Drug research is ancient, and in the Vedic books like Sasruta Samhita, written in Hindi in Sanskrit language about 1200 BC, and Chakrashringa 300 BC, and also at that time Takshashila University 5th century BC, Nalanda University 5th century AD, actually very much prominent. But all those things, what happened during the Muslim invasion between the 700 to 800 AD, ruined the Hindu dynasty. British would then ruin the Muslim industry dynasty, but again banned research on Phyto-Drugs because at that time during the 1926 to 1960, penicillin, streptomycin, erythromycin, all drugs was developed from fermentation of and easily available. So later on, some Phyto-Drug like curcumin, quinine, artemisins, Taxol was used by different workers outside India, and those are very good Phyto-Drug. And recently India, some activity is going on because Herbal Institute was made and government giving a lot of money. Here my extraction system, you see 15 ml tube, that some small cube of the extract of the plant part, and because the tubes is made in Germany, so very stable and no activity of leaching, that's why I use this small 50 ml tube. And then we make the different part like fruit, leaves, bark, root, flower, and then make the extract. They are different color. You look leaves, flower, fruit, bark, root, and then we assay, Kirby-Boyer assay. You see this is imipenem, very good to inhibit MDR bacteria because most bacteria are imipenem sensitive, and those are imipenem resistant. They are terrific bug. They are very much bad. But you see the root is the greatest activity than bark, and ampicillin is here, and flower, leaves, fruit have some little activity. Those are not good because those are not 15 millimolar. And we did the TLC in an 18 into 15 centimeter plate, and we use this type of jar, 4 jar, 2 in each jar, and here is the dried, and you see that this is the one of the good potential which we characterize as a RNA polymerase inhibitor and good antibacterial activity, and which I am giving later bit. But what is the story here? Summary, that we made some MDR cure from 5 or 6 extract, and they kill the MDR nail infection and also MDR infection in rats. And these, within these 6, actually Suragada multiflora, Josepha gossypilia, and Cassia fistula were greatest because their activity concentrated in a zone I can be separable through the TLC and could be commercially explored for further characterization. So this is a E. coli KT1 MDR bacteria we isolated from Ganga river water, there is flagella and one spore, and we used for characterization. Here is the drugs. Here is the some activity statistic in the USA. They shows with the time more drug resistant bacteria isolated more in New York area, and this is the 2010 study. So this is actually now spread all through, and but this study I have not with me, and most of the carbapenem and cephalosporin both are, and ampicillin is totally resistant. So this is the some activity where antibiotic could be phosphorylated or acetylated by enzyme we called MDR gene, and the arrow indicated the site for streptomycin. And this assay to see if we extract a MDR bacteria extract and put on radioactive acetylene, they acetylate, and those acetylated chloramphenicol move very fast. This is the normal chloramphenicol. Acetylated chloramphenicol is more molecular weight, but still they move very fast. And you see 1,3-diacetylated chloramphenicol very fast, near the upper panel. So that is the good assay. You can do it. And here is my first assay. We showed when I put the different drug and keep the plate more than overnight, the drugs, some zone is appear inhibition by the antibiotic, but it is filling up with time. So that means both in Ganga River water, both resistant sensitive bacteria, and when we plated, then minus drug, so many bacteria, then ampicillin 50, still more bacteria of tetracycline. Then if we use the tetracycline and chloramphenicol, then it is reduced. This shows here the number of bacterial colony was increasing with different and antibiotics, and imipenem is 0. But we got imipenem resistant, but we have to assay the 10 ml water, 10 ml water. So this is the action of ampicillin resistant, amoxicillin resistant, cefotaxime resistant in the drain of Kolkata water, rain water at Kolkata, and Ganga River water. And you see the cefotaxime bacteria is less, and cefotaxime is a third generation, but with imipenem 0. Here is the some resistant bacteria profile antibiotic plate. You see those are resistant because no clear zone. Here is the imipenem, but E. coli DH5 alpha, which is genetically make no plasmid there. So all antibiotic is susceptible. We made the 16N RNA gene sequencing to know which is E. coli, which is Pseudomonas by blast analysis chart. This is the sequencing data. And we also got some drug-resistant bacteria from phenalkaligen and Stenotrophomonas, which are sometimes called. We see the here reaction of the MDR gene, AMP gene, everything is here, all bacteria has. Then CTXM, few bacteria has, but few not. Here, these and these lane not. And also acetylating gene, AAC gene, there is in some, but not in one. So it is abundant. And this is the individual gel. This is the primer, and this is some PET and ACR drug reflux gene, which also present in the KT1 bacteria. So when that bacteria is inhibiting, that means drug reflux efflux genes also inhibited. This is the SSN number we deposited. And then this is the plasmid from the chicken meat and human hair, some bacteria which is resistant to ampicillin, tetracycline, chloramphenicol, streptomycin, ciprofloxacin, and azithromycin. We see 100 kb bacteria also there. Now, why we are not showing the plasmid profile of KT1 E. coli from Ganga River water? Because they give smear, there is so many plasmid, and confusion was resolved, but I am not showing here. Also, milk contains some bacteria, but pasteurized milk, no. This is the cow we studied. And then this is the one of the tree that blossoms. This is called golden sour tree, and this is in our school campus. This is our, looking our campus gate. And we use the bark. Here is the flower. Here is the pod used to dance the monkey. So that it is Madorlati in our local language. It is very much abundant in India and Thailand. These are the some TLC, and this is CU1. It is can be visible by eye, naked eye. So you can cut the band. This is the assay system. So it is more than 15 millimolar. And here you see the TLC plate. This is the, we cut this band, but when we put the UV, this is another fluorescence band. It is strong. And when we cut this band and acid, it has strong antibacterial activity. So this and CU1 and CU3 both have antibacterial activity. This is the HPLC. It's eluted very quickly. There is some contamination, possibly some turpentine. The assay of the biochemical assay, but the important thing is the elementary detection analysis. Carbon is 35%, hydrogen is 5%, and nitrogen is 0.03%, means no nitrogen. Then oxygen is 59%, that is impossible. No compound is there with in plant. So could be halogen derivative. We consider our bromine or chlorine, and ultimately we see here the mass spectra where you see these 6 band, all the difference is 82. So that is HDBr. And here cut value is 100. So we cannot see the Br minus. But when we see the other one, here is the two isotope, 79 and 81, and that 79.5. You see here some two close band. So that means bromine atom, 6 bromine atom. We did some NMR study and actually FTIR study, NMR study, and also carbon NMR, that is proton NMR. And when we do the individual study, we see the OH group at 300 to 34 centimeter minus, and here is the bromine atom, and here also the bromine atom for CBr, and this is the some polyphenolic compound we suspect here. It is all data. Here also we see some of the activity, and that shows the bromine atom and also OH group. And this is here carbon NMR. We found here CBr, but here also CH should come. So this is the broadband, and here is small band that is should be bigger, but I don't know why, and that is COH, CO bond. So we pretty confirm, but I actually don't know the two-dimensional NMR, so it is not done, and structure is incomplete. So we send 11 compound TLC made to one of the laboratory for RNA polymerase activity. So this is the rifampicin. This is one of the compound NU2, and this is CU1. So this is comparable to rifampicin. We characterize this, and where you see that E. coli and also Mycobacterium RNA polymerase was inhibited. And this is the confirmatory plasmid-based run-off transcription. We found 70 nucleotide run-off transcription, which gradually diminished in presence of CU1 at up to 160 microgram per ml. So it is confirmed the RNA polymerase inhibition. Further confirm is come when we shows the EMSA assay, and we see the protein DNA, radioactive DNA binding, which also gradually diminishes with increasing, and no at all at 60 microgram per ml of CU1. So it is confirmed, but DNA polymerase activity with increasing up to 160 microgram per ml, here you see no, all equal, no activity diminished. So DNA polymerase was not. So it is very specific for RNA polymerase. So now we coming to the other plant which known as Suragada multiflora or Naringa. The history is this plant used to treat polyjor or malaria in the ancient time by my grandfather. So I sent some extract to John Hopkins University School of Medicine, USA, and they said 69% inhibition of, and later on I joined there and did some work. Here is some of the activity on E. coli, Bacillus subtilis, and Esodias. The inhibition as is greater than 15 millimolar. And here is the Suricata multiflora UV fluorescence band that 4 times purified and runs very fast. But initially, such gel, this band ran in the middle. So when we purified the garbage, they run very fast. And this band is cut, and then NMR, FTAR, biochemical assay was done. So that's why the amount of phytochemical and one particular type phytochemical is important and in our system. And when I John Hopkins, we see some malaria component proved it. So here we see that some malaria parasite inside the O plus human RBC, and this is the malaria parasite fluorescence, and they inhibited actually with the drugs. This is the parasite, these are the inhibition. So we confirmed that malaria parasite indeed very inhibitory. We send this drug to Indian Institute of Chemical Biology, my PhD supervisor H.K. Mojumdar, and they did Leishmania assay, MTT assay. They also shows this activity inhibit Leishmania dodomani. And this is some of the inhibition of Trypanosoma brucei, just in vitro assay. They inhibited also the activity of. So this is the mouse 3T3 cell, eukaryotic 3T3 cell. They are less inhibited up to 20 microgram per ml in cell culture. This is some of the actually NMR spectra that we did. And they shows that is glycoside. And finally, the RNA polymerase assay we previously did, there is little activity of RNA polymerase activity, but when we did in E. coli, but when we did with Mycobacterium RNA polymerase, that is not inhibited. So we forget about the RNA polymerase inhibition. We purchased some E. coli DNA polymerase 1 and we assay the different. And here is Atropa inhibition, J2, J3, NU3, NU2 is inhibited here. That is the inhibition. What is the assay? Supercoil plasmid by topoisomerase should be reduced to topological isomer here. And this is the relax band. Could be the supercoil relax, circular relax. But these assay you cannot understand. Here we can understand, but these are the topological isomer. This is the topological isomer. Well, we run a little bit higher mobility. You have to run 20 volt for 12 hours, but now my lab, I cannot stay at 9. So I forgot I did the 6 hour assay. Now we did the 1 microgram plasmid DNA here. This is supercoil. Now with enzyme, the relax. And now when we less enzyme, this is the topological isomer. We relax less and less, and then ultimate relax here. But when we add 3 different concentration of the drug NU2, we see they can and totally inhibit at 20 microgram per ml again. This is the lambda Henry 3 marker. And so we prove that definitely this has topoisomerase 1 activity, but the corona infection and our lab closed for 2 years, everything gone. But I am now publishing this parasite Leishmania has the mitochondrial DNA. This is the present in the actually bugs, and this is present in the actually. But those are very important system to show the inhibition by topoisomerase. Here, when I add the topo2, they are linear, small circle, and few maxi circle not shown here. Here, supercoil DNA, they relax here. This is my PhD thesis. So I did some assay on the nail of supercoil infection, those not, and they are cured. And we treat some rat with the MDR KT1 and then inject the drug and then antibiotic. Cefotaxime did not clear, which is third generation antibiotic, very strong. but that superbugs was eliminated by phytoextract 0.5 ml, and not completely, but 83% inhibition. We acid the blood till puncture blood. And this is the other tree which has some potential, the bark of Jatropha gossiphilia. The story is, in the boyhood, we used to brush our teeth with this tree and no harmful at all. We know that is no toxic to human. So I show this picture before. So this is root is very potential, and here is that TLC assay, and here is the comparison, and here is the two spice which has very potential. One is cinnamon, other is cloves. And worldwide, those two use, those has very potential, but they have no specific concentrated actually chemical which we can commercially do very well. Similarly, we study some rose or different flower actually. Here I show some picture interesting. This is the rose band and this is the yellow band of marigold band, phytochemical. Those are not inhibitor at all. Some activity we see, but when we purified these and assay, no activity. But they do very color change with sodium hydroxide, and then in green color and in HCl, so they are deuteriode. But we can use to as a abir or some festival in our country, and then we can actually make clothes. Here is red and green and blue or green, but this have some kind of activity. In conclusion, we have identified few phytochemicals that are useful to make MDR lotion against MDR bacteria. C1 turpentine polybromophenol was purified having RNA polymerase target, which may be the useful anti-TB drug. NO2 glycoside was purified with DNA topoisomerase 1 target and may be useful against malaria. Further study necessary to PONC and other target and structure prediction. But I am retired, and thanks to COVID-19, everything spoiled worldwide. This is some publication, and finally we got one Elsevier publication, current research in pharmacology and drug discovery in 2022. That is one good, but otherwise small journal. And one is also Indian Journal of Biotechnology. That's also good journal. But thank you for your time. And that is the end, madam.